Hello, and thank you so much for the opportunity to talk to you all today. It's an absolute pleasure to be invited to talk at the CBTN event. It's currently nearly 2 o'clock in the morning, so forgive me for recording this. My name is Mark Cowley. I'm a computational biologist at the Children's Cancer Institute, and today I'm going to be talking about the Australian Bioinformatics Data Commons Pathfinder project. I speak today, as always, on behalf of a great team of people, and this particular project was a great collaboration with Andrew and Stephen and many others from the Australian Biocommons, Alison and Adam from CHOP, who you all know very well, Jack from Seven Bridges, this was a great industry collaboration, and Marie and Chelsea from my team over in Sydney, Australia. To give you a bit of a framing from where I'm coming from, I'm fortunate to represent the Zero Childhood Cancer program today, which is Australia's first and largest paediatric cancer precision medicine program. We launched back in about 2015 with a pilot study that attracted nearly 60 children, and then we launched the national program, which has been quite a great success. All 8 paediatric cancer treating hospitals around the country recruit children to the program. We've recruited well over 450 children, and at this stage of the program, we've only been recruiting children with very high-risk cancers, those that have got less than 30% chance of surviving 5 years. So it's a very heterogeneous cohort, about 30% brain tumours, 30 to 40% sarcomas, and a range of mostly relapsed and progressive disease of other types of solid tumours and leukemias. Our approach back then was probably a little unique in taking a deep whole genome sequencing approach, as well as an RNA sequencing approach, methylation profiling, originally just for the brain tumours, but now for all patients. And where possible, we do high throughput drug screening and PDX modelling of patient tumour models, and we work very, very closely with our clinical colleagues around the country. Zero is a collaboration between a research organisation, Children's Cancer Institute, and the Kids Cancer Centre, our clinical partners up the road, also in Sydney. We're very fortunate to have been funded to expand the program ultimately to every single child in the country, low risk, medium risk, high risk, and if they relapse, they get to be re-enrolled and get repeat molecular profiling, so that by about 2023, all roughly 1,000 children that are diagnosed with cancer in Australia could become enrolled. So it's an incredible program to be part of. Really, this is the one data slide of what we've done, is that we recently published our experience from the first nearly 250 patients. And these numbers, which I won't have time to go into much detail, really just reinforces the very high rate of detecting variants when you use such a comprehensive platform. Just to pick 2 of the numbers out of there, over 70% of children received a targeted treatment recommendation when we applied this comprehensive molecular profiling approach. And on the far right, and I think my face is covering the numbers, but of the children that received the recommended targeted therapy, 71% of them had a clinically meaningful response. Half of those with stabilisation of their disease, and the other half with shrinkage, all the way down to a number of patients with a complete response. So again, pretty exciting stuff, and I think that helps frame really the point of view that I'm coming from today. And really now I would like to tell you a lot more about the Oz Biocommons Pathfinder project. All 8 of the treating hospitals that are referring children into our program are indicated there with red dots throughout the country. And through their collaboration, we're able to recruit almost all of the 200 new cases of high-risk paediatric cancer each year. Fortunately, that number is not larger than it is, but it's small enough that we need to really think about aggregating data so that we can accelerate research into the understanding of the causes and treatments for high-risk kids with high-risk cancers. It's also really important to us that we share data with our Australian research and clinician partners much more easily than we have been able to before. So these are some of the things that we really wanted to try and tackle. So you can probably tell what's coming up next, given CBTN, and I guess I still remember hearing Adam Resnick talk for the first time back in 2019, and it really was inspiring. And so what we tried to do is kind of frame a lot of engineering in a simple scientific question. which is can we share, combine and integrate brain cancer data from local and international patients to hopefully discover new disease subtypes. We haven't quite done the last bit, but we've done almost everything else, and I'd love to tell you about what we've been able to do in We split the Biocommons Pathfinder project into 6 different work streams, from creating workspaces in AWS Sydney to wrapping and optimizing our bioinformatics workflows, bulk processing of the Zero and the CBTN datasets, performing an interactive analysis of the brain tumour transcriptome data, and integrating this infrastructure with our local infrastructure. In this particular work stream, the Seven Bridges team did such a good job in a couple of months bringing the Cavatica platform to Australia. It felt like an extremely seamless process, and now we're able to create projects that are in Asia Pacific Southeast, which is the same data center where our data actually lives. So that just seamlessly works. So in the interest of time, I'll just move on to the next one. In Workstream 2, we wanted to wrap and optimize our bioinformatics workflows. We'd already been using the cloud for 5 or 6 years, and so felt fairly comfortable there. But we felt like our pipeline, cloud-based pipeline, only ran in one cloud, and we really wanted to take advantage of multi-cloud technologies. So we borrowed over 20 existing tools or Docker containers, Dockerized another 25 or so different apps to take care of different parts of the pipeline, including 3 of the tools that we use for our precision medicine variant interpretation workflow, including Helium, which helps rise the pathogenic variants to the top of the list. Many of the tools are fairly common in the pipeline, but I want to shout out to our partners at the Hartwig Medical Foundation, who've written some extremely good somatic cancer analysis tools, SAGE for SNVs and indels, and then Amber, Cobalt and Purple, which work together to give excellent somatic copy number analysis capabilities, as well as Grids 2, which is an excellent structural variant calling tool built by Daniel Cameron and Tony Pappenfuss in Melbourne, in Australia. I'm not sure how many people are familiar with running tools on the Cavatica pipeline, but you get these beautiful representations of how the pipelines look. The green circles are the nodes, the jobs that do things, and all the grey circles are the inputs or the outputs. And you can quite elegantly see how even a pipeline here, this is a small part of it with maybe a dozen steps, has many dozen inputs, outputs chained together in very specific ways. As I said, we took the opportunity to optimize everything and determine what the resource requirements were, and we were committed to adding some features along the way, which is BAM and CRAM support, multi-cloud support, and importantly, switching over to the latest reference genome. Not quite there, we're doing end-to-end testing and pushing ourselves to have that all finished by the end of June, and all using CWL. So that's been a great success. As further evidence of the success of that part of the process, if we look at the comparison of how long the pipeline takes to run and how much it costs, when we compare to our previous cloud-based pipeline, the time to take an end-to-end, so when I mean end-to-end, it's sort of fast queue with some quality control all the way through to variants, annotated variants. The time reduced by about 4 hours when we use Cavatica, which is 18%, but remarkably the cost reduced nearly 80%. And that's in part due to some of the optimizations, but really in part to being able to use the spot market. That was a really significant and impressive saving. I'll talk a bit later on about integration with our environment, but if we have our data stored on a NetApp disk array, it takes a little bit longer. It doesn't cost much more, but works extremely well. Essentially, whether you have your data sitting inside the Cavatica environment or whether you have it sitting in your local on-premise storage. Certainly, we're pretty comfortable with the cost benefit. I'll talk more about that in a second. So we feel like that was a major success as well. I should point out this is quite deep tumour normal sequencing. So this is minimum 90x, often almost 120x depth sequencing. So and the full analysis, SNPs, indels, copy number, and structural variation. So quite impressed with the performance of the pipeline. Love to sneak it to just under 24 hours. The pipeline I just talked about is the whole genome pipeline, which is the more complicated one for us. But when we talk about analysing data in this pilot project, we looked at RNA sequencing as we felt that was a more lower risk and more attainable goal. So in work streams 3 and 4, we wanted to bulk process zero childhood cancer data and the CBTN data sets. So essentially we had nearly 1,000 brain tumour patients from CBTN with RNA sequencing data and over 100 patients from zero childhood cancer in Australia. And fortunately, we didn't have to reprocess the CBTN data because they had already run it through their great RNA sequencing pipeline, which was pretty similar to our own. And so the goal was to run the 112 Australian samples through the Kids First pipeline. And I think we take it for granted now, the idea of bringing the algorithm to the data. Gone are the days where you try and download all of the data to your data center and run it. So essentially what we were able to do is take that Kids First RNA sequencing workflow shown on the right and run it through our 112 patients, and then only copy a small amount of process data, the gene expression data, into a workspace and perform an interactive analysis. So that was quite a pleasure. I think within under 2 days, Chelsea had been able to process all the RNA sequencing data through those pipelines and perform a clustering analysis. Everyone that's run RNA sequencing analysis and tried to merge 2 data sets together know that batch effects are really important to try and model and correct. And that's compounded by the Zero team using mRNA sequencing, whereas most of the CBTN data is actually total RNA sequencing data. So we did some Chelsea in our group did some pretty heroic batch correction to try and make those data more comparable. And I think we're a smidge disappointed at this stage with the separation of the clusters between these different patients. We're still mining this data to try and find really interesting clusters. But on the right, you can see when you annotate by the cancer type, there's a really large cloud of low-grade gliomas in the top, which are predominantly from CBTN, because most of the patients on the Zero study are actually high-grade gliomas. And I think at this stage, given that we want to reprocess all of our data with total RNA sequencing, we haven't pursued finalising this analysis as aggressively as we might have, because we know a whole bunch more data is coming, and then we'll pick up this project to try and find those new subtypes. The last slide really indicates that we've been able to integrate Cavatica with our local infrastructure. So we actually use a data centre for most of our storage in Sydney. It's the same data centre where the Amazon cloud also lives. And so we were able to use a NetApp grid engine to end up storing our data in there. And then we can expose that data via an S3 bucket straight into it and mount straight into the Cavatica platform. We felt that this was a good way to go because I think everyone who's used the cloud knows once you put a piece of data up there, it can live there for a few years without being touched. And so if you can, which of course you pay by the month. So we felt like this was a good hybrid arrangement of having data locally and then exposing it seamlessly into the Cavatica workspace. And so that worked really well. Our partners at the National Compute Infrastructure have had a similar success doing. So the take-home messages really are that the Cavatica platform is now running in Amazon Sydney. We've been able to develop multi-cloud and connected cloud storage as key components of our analytical workflows, enabling us to collaborate with partners in America. From an Australian perspective, this has enabled us to create one of the largest paediatric cancer data resources generated in the country, boosting our numbers almost tenfold immediately, and lots more collaboration there to explore what we can get out of that data. Our graphene workflow will be available soon, and we're trying to publish that as well. And really what's next for us is trying to make our data more fair and more findable and reusable, etc. So we're building a Gen 3 powered web user interface, focusing on security, passports, access, authentication, clinical metadata, and how we might further integrate data from Zero and other Australian partners, including the University of Melbourne, and the Australian Genomics Health Alliance, in part funded by an ARDC-funded grant. Thank you so much to the team that worked on this project, especially Alison, Adam, Miguel, Yankun and Bailey from CHOP, our group over in Children's Cancer Institute, the team at Biocommons, the Australian Research Data Commons team, NCI, ARNet, Bioplatforms and Seven Bridges. With that, thank you for